you can use this form to design primers for PCR-based cloning in yeast. You can use it for design primers for gene deletion, C-terminal tagging, promoter insertion with and without tags. These are cloning strategies that look like this. You can see an example in OpenCloning. So there are strategies that use a plasmid. They amplify a part of that plasmid using PCR and adding homology arms to the PCR products in the primers, and then those homology arms enable that piece to integrate into the genome and modify it to generate an allele, in this case a deletion. So let's see how to use it. Here you have some instructions that you can read on how it works, but I think the best way to get started is to just run an example. So let's pick Pombi as the species, let's pick gene deletion, and then we introduce a couple of genes by their unique identifier, and then let's pick the option design primers only. So when we click here, this is gonna design some primers for performing the cloning and for checking. So these are primers to amplify from the plasmid, and these are the checking primers that sit upstream and downstream of the gene that you can use to verify the insertion is correct. There is the option here to edit the sequence of the primers that binds to the plasmid. So for example, this bit here that binds to the plasmid to amplify the cassette, you can change it here if you're using a different plasmid, or if you prefer to use different TM or something like that. Now, what happens if we select design primers and simulate cloning? In this case, we have to specify the plasmid that we're going to use, so we can use the default ones in some options. So in this case, it will be the PFA6A kanamycin MX6, and then here we specify the resistance marker, and similarly we can specify checking primers. So these primers are the ones that will bind to the kanamycin resistance cassettes in either orientation for checking that the insertion is correct. So we can then click and submit, and then after a small delay we're going to get a zip file in our computer. So we got it, and if we unzip it, what we'll find inside is a summary Excel file that we can open in Excel. And what's interesting here, for example, is that we can see the same primers that we saw before in the web form, but we get some extra information as well, because we see that in this primer the part that binds is not only the one in uppercase, or also the A binds, and the same with this primer here. It's not only this part with the uppercase that binds to the template, also GAT binds, so that alters the TM. So this can be useful for you if you care about PCR details a lot. Then if we go to the folders named after the gene names, we can see that we have GenBank files for a bunch of sequences, and this file called cloningstrategy.json. So this is a file we can open in OpenCloning, so we can just basically drop this file in here, and then we will see all the steps that we followed to make this allele. So we see the plasmid, the fact that we've done a PCR from it, the locus we targeted, and then here the modified allele. And then on top of it we have the PCR for checking the insertion. In this case is the left one, and if you click on this eye and see the sequence in detail, you can see where those primers used for the checking are bound. So you can see them here. You know, this is the checking primer on the left side, and then on the right side is just here. And that's it. There's other options you can change, so you can provide a plasmid from a gene. If you put the identifier, you can upload your own plasmid, but basically you're ready to start. If you have some feedback, please reach out to the Baller Lab. Bye.